Hey guys, in this video we'll be going over basic bacteria identification, their shape, and a process known as gram stains. To start off, let's look at the circular-like table before us. Each box shows a drawing of what we might see under a microscope if we were looking at some bacteria. Going down vertically in the table, we see that on the left all the bacteria are in a purple color, while the right has pink-colored bacteria. Going horizontally, we see that the top row has a spherical-shaped bacteria, while the bottom row has rod-shaped bacteria. All of these characteristics that we have identified tell us something specific about our bacteria and even has a name for it. To start off, the shapes that we identified before, sphere and rod, have their own specialized names. The singular version of our spherical bacteria are called coccus, with a plural being cocci. A single rod is called bacillus, while multiple rods are known as bacilli. These are two of the three main shapes that bacteria can take, with a third being a spirochete, with multiple called spirilla, and they take a squiggle-like shape. Now the color of that bacteria comes from a special stain called a gram stain. The purple-colored bacteria is what we call gram-positive, while the pink is called gram-negative. Now the reason why such bacteria even have a color is due to their structure. In a gram-positive bacteria, we have four important layers that surround it: the plasma membrane that encloses the bacteria's cytoplasm, the periplasmic space, a thick peptidoglycan layer, and the capsule that is the outer envelope of the bacteria. In a gram-negative bacteria, we have more layers surrounding the bacteria, with the plasma membrane being split into the inner and outer membranes, and the now thin peptidoglycan layer, along with the periplasmic space, being wedged between the two membranes. Outside the outer membrane is the LPS, or lipopolysaccharide layer, with the bacteria's capsule following suit. Looking at the layers of the gram-negative and positive bacteria, we can see the peptidoglycan layers from gram-positive are disproportionately larger than gram-negatives, even though the collective height or thickness of the layers are the same, due to the gram-negative bacteria having more layers. This difference in thickness for peptidoglycan is what is key in the bacteria being stained purple or pink. The gram stain itself leaves a purple color and will stick to the thick peptidoglycan layer of a gram-positive bacteria, but will wash right off the gram-negative's thin peptidoglycan layer. Later, the gram-negative bacteria will be counterstained by safranin, which helps get its pink color. I hope this video helped you to understand bacteria's shape and structure and how we can determine its structure through gram stains.